Okay.
Okay, I think we are live. Speed of sound here. I am old and irrelevant, so I'm going to take a back seat to this discussion. But I have the remarkable Aron Ra and the remarkable Dimiter Kunnev for a discussion on abiogenesis, and we'll see what they end up doing with it. So, over to you, Aron. What do you got for us?
Did you say over to me, Aron?
Huh? What's that?
Were you calling on me to speak first?
Oh, I did. Yes. Is that okay?
Oh, yes, absolutely.
Yeah.
You had mentioned that you had 4 of my videos on abiogenesis in the description. I wasn't even aware that I made 4. I'm going to have to think about that.
Well,
I do have extensive notes on different studies for different aspects of evolution. And one of the things that I realize that when people don't understand it is that they expect that you're gonna pour in like 3 or 4 bottles of household cleaner into a machine and it's going to crank out a rabbit. And I don't understand that it's a hell of a lot more complicated than that. Way too complex to be explained away from somebody saying abracadabra and poof.
Right.
And poof.
Yeah.
So,
So one of the things that I have in this list is like how we have a chemical pathway to ATP synthase in the study that supports that, and another one that shows how we have the phospholipid bilayer automatically forms because of chemical polarity with water, and then we have how montmorillonite clay affects the autosynthesis of RNA nucleotides and blah blah blah. All these different stages that people have to understand what's one step at a time. And there's different processes. A lot of people confuse this with evolution. Evolution is a single process, a genetic process, and abiogenesis is a collection of processes in different chemical environments. And so you can't confuse it is necessary to have a first life in order for evolution to begin. Evolution starts with life. It's not the origin of life, which is another thing that people don't understand. And I understand that you have to have the origin of life in order to have a comprehensive explanation. But there's no way around it. Abiogenesis is a different process or a collection of processes.
Yeah.
So I'm excited to hear Dimiter give yet another element that I can put into my list of different scientific studies supporting abiogenesis processes.
Yeah.
And this one is at the very, very beginning. And I don't know if you saw that last little email I sent out, but I'm very, very adamant about defining the very, very early steps and the understanding that there are steps and every step has its own probability and stuff. And I thought I did a good job. We're going to be doing more videos on that, but go ahead, Dimiter.
So, Aron is correct, actually. There's right now 2 major approaches to origin of life. One, it's called top-bottom approach, from top to the bottom, and another approach is called from bottom to the top. So these 2 approaches have to meet somewhere in the middle. Speaking from top to the bottom, you just try to see what kind of structures, biological structures and cellular organelles, for example, are necessary if you simplify everything necessary in order life to exist. And that's the top-bottom approach. And usually in that case, you finish with 2 processes, basically replication and translation. That's the bottom of the entire row. You go from the top to the bottom, and you finish to replication and translation as 2 necessary small processes that you need to have in order life to start, to kick out. From the bottom to the top type of approach, you of course have a huge army of chemists which are working all the time, and recently has been shown from the laboratory of John Sutherland and many other laboratories, 1 pot or 2 pots or several pots of reactions, that nucleotides are quite possible. So all the building blocks of life are absolutely feasible to be synthesized. And there is too many papers for this. We don't have time right now to go over the chemical part of the everything, but I hope I will be able to explain pretty clearly how the entire system is assembled in a way to start the life process. So this is actually the more difficult part, is the more unknown part in the origin of life research. And this is what the people usually are very confused for, because how, if you have just blocks of life, something will start and organize itself and make successful selection process. So that's why I would like to just start over. So I will put my slides, and let
me
keep going.
I'll share you.
Let's see. Here we go.
Oh, there you are. Gotcha. Is that big enough, or should I get rid of the side? Get rid of our cameras too. It's up to you.
I
think that looks. Yeah, it looks
well
good.
Visible. Still
it
well
looks good.
visible.
Okay. Go ahead.
So, also, just a few things I have to say before starting everything about myself, just to introduce myself. So, I'm a molecular biologist. So I was working experimental molecular biology in a cancer research for probably approximately 20 years. And origin of life, it's not my primary research interest, simply because there is no funding for origin of life. And it's a very little funding, I have to say, for origin of life. But also I stumbled, so I entered in this topic quite kind of unexpected for me, because everything started approximately maybe 5-6 years ago, when I actually debated with creationists for evolution versus creationism. And usually those kinds of, everybody knows, those kinds of debates come to origin of life, of course. So I'm not going in details, definitely, because we don't have time, but such a conversation actually triggered in me to think about. And after a relatively long time, maybe 1 or 2 years of thinking how can happen, and so on, I came up with hypothesis, which I will describe it today here. But this hypothesis actually, it's to me, it was very well explaining. It was capable to explain almost all the questions in origin of life. I mean, all the questions mean principal questions in origin of life. And what was also very interesting, when I was in a process of develop my hypothesis, I didn't read anything from what actually the scientists are about origin of life, and how the all the professional in the field of origin of life actually was thinking about origin of life. So pretty much I was familiar with RNA world hypothesis, but nothing much. And when I started to become familiar with the published material, with the published papers, and to see what the people that someone is paying to develop and to work on origin of life, I was completely shocked to see that whatever it was in my mind as a hypothesis actually was not in the literature at all, and was much more different things. And those different things didn't make much sense to me in many ways. So I will explain to you, of course, today what I mean exactly. But it's suddenly, so I read probably about 100 200 papers for origin of life, just expecting to see my hypothesis somehow formulated in one way or another, with different words, of course. But no, it was not there. So it seems to me that somehow I was trapped in my own hypothesis. So I said, okay, so I have something here interesting, definitely can explain a lot, and it's not published. So in that case, I decided, okay, let's go and publish all of that. So I published 2 papers. One is 2018 paper, and the another is 2020, this year, paper. And I'm preparing right now the 3rd paper, which is, I will submit it probably until the end of this month. So the first part of this today discussion, the day presentation, will be mostly about this last paper, the last 3rd paper, which is not published, but it discussed mostly things about what we can call life, how we should looking for the origin of life, what is the most important thing when you just go over and try to think about how life originated. So that was kind of my journey in the origin of life. So let's keep going. See the next slide. So what I can say here, in order one hypothesis actually to be a reliable, good hypothesis for origin of life, it needs to give answers, at least possibility, feasible possibility, pathways which can describe all of this and give an answer to all of these questions that I'm listing here. For example, a good hypothesis for origin life should not have too much gaps. Meaning, if you describe something that it's starting from here and goes there, you should not have too much assumptions about that.
I would like to jump in on something if I can.
Yeah.
When you say that you're talking about possibilities, and remember we're talking to an audience that very often, or very many of them, don't really adequately understand science, and and a lot of these people think that anything is possible, that it's possible for monkeys to fly out of your ass and for a cow to jump over the moon. So when science identifies a possibility, they're talking about chemistry that actually does work, not just could have worked, but the way that we know that it could have worked is because that it does work in chemical experiments today. So, and then I'll save the rest of my interruption.
So that was important to note. A lack of gaps mean you should not have something that start with and suddenly assume that many steps happened with unknown how it's happened, and just continue to explain the rest of the story. So a lack of those kinds of gaps, which is very important for one good, reliable hypothesis for origin of life. Also, origin of life needs to solve the chicken and egg dilemma. Everybody knows about the RNA or proteins comes first dilemma. So those kinds of thinking was at the very beginning, back in the 80s or even 70s, how it happened. So it must be solve this chicken or egg dilemma. Another thing, it needs evolutionary justification of how the translation form. Meaning, what does it mean? You need to explain what kind of selection forces lead it to formation of translation. Because the current biology that we know of, it's based on translation and replication, of course. And that's a very important. Another question is how the standard genetic code come to be. And this is linked immediately with the question how the information's kicked out. And that's absolutely important to understand that information must appear spontaneously, and you have to explain how that happened. And it's a question, a real question. So origin of life, it's about origin of genetic code as well, and origin of information as well. There's another question for a very good hypothesis, like how the ribozymes was naturally selected. And this has come to the problem because ribozymes are enzymes which are entirely built up with from RNA. There is no protein in the ribozymes. However, as enzymes, they in competition with the protein enzymes as well. So there must be a selection force which somehow explain why ribozymes should be selected anyway, because at the same time you can select proteins for the same purpose. So, and that explanation also is very much debating in the literature. And if you go in over the papers, you will see lots of trials to explain this particular dilemma. Needs explanation what kind of simple structure will start the life. So what I mean here, if you think about the records for origin of life, I mean for life, our very earliest possible records, they go back to the 3.7 billion years ago, even further. So in order to life to actually the time when life began was almost immediately from geological perspective after Earth, when Earth actually chilled down.
We also need to specify that when you're talking about the very earliest life, that we are we're talking about microfossils of bacteria dating to so far 3.77 billion years back. But that's still bacteria. And that when we're talking about in the origin of life, we're not talking about dead chemicals suddenly, that there's not ever a lightning strike in a mud puddle and suddenly poof, anything is alive. It's not like that. So when you say that starting life, a lot of people will get the impression that you have, dead chemicals in a puddle and then suddenly they're walking around. So
that's what I mean. Yeah, exactly. So that's what I mean as well. So
there are transitional species even in the origin of life. So like viruses, for example, are technically not alive because they don't meet all of the criteria to be alive. Absolutely. Yet they do evolve and they can be killed. So they're kind of like undead. But they're also similar to the hypothesized protobionts, which is one of the transitions between from replicative polymers to another hypothesized hypercycle, which is like another prior cell.
Yes.
And these protobionts are very much like viruses. And so we don't know that if modern viruses are a stem from a protobiont or if they're a degradation from a prior form. And I'm sorry to interrupt again. I'm clarifying for the audience where I think they need to, and they might also need to understand the difference between or what an enzyme is as opposed to what a ribozyme is. I don't know. I didn't see any questions about that in the chat, so I'll leave it alone.
So the ribozyme is an enzyme that it's built entirely. I mean, it is assembled entirely from RNA. There is no protein part there. That is a ribozyme. In contrary, the enzyme is actually a protein. So, and this is the difference. It's very important to be explained the selection forces, which somehow, because there is a natural ribozymes, and you have to explain how those came to be. And this is one of the major kind of clue that started and give rise to the RNA world hypothesis, but we'll talk about that later. So my point was here that the actual data that suggests so early origin of life to happen, meaning that you expect something which is kind of very possible to happen. Statistically should be very easy to happen. Otherwise, you can imagine another scenario, Aron. For example, another scenario could be just imaginary scenario that there is no any traces, fossil traces, either microfossils or whatever, around 1 billion years after the Earth formation. You can imagine those kinds of scenario as well. In that case, maybe panspermia might be more reliable answer, or some extremely rare event to happen. So it might be a little bit more plausible answer if that was the case. But this is not the case. I mean, the microfossils goes too back to the beginning of Earth. So we need our explanation to be extremely simple and extremely possible, statistically possible. So this is also very important. And of course, everybody, so usually us molecular biologists blamed always when we are talking for origin of life that we don't care about the energy, where the energy came from. And this is usually the blame which is coming from the metabolism first origin of life advocates. So, of course, it needs to explain how the energy comes as well. So, speaking of this, so what else I can say here? So that's kind of everything that I wanted to say. So let's go for the next slide. So here, this slide is extremely important. And this is now the slide which will explain a lot when you're thinking about what actually we are looking for during the our research for origin of life. There is probably around 200 or maybe more explanations, and they definitions for what life is. So the goal here, it's not to give just general definition of what life is. The idea here is to give us such a definition which can go deep into the bolts and nuts of the life itself, what the life is to exist itself. And it's all about the Darwinian evolution. It's the formation of Darwinian evolution. And here I can describe, so the system, a very simple system, when you have some sort of information, source of information, which happen to determine somehow by a specific rule, a structure or some function. And later on, I will describe, of course, based on the hypothesis, how that happened and what is the actual structures and functions. But right now, just in general, to see the major components of Darwinian evolution. If this information determine some structure which possessed a function, and this function actually support or somehow let me say, make more stable the informational source, you have a positive feedback. So if you put this system in the environment, what will happen? It will happen, two things basically could happen. Either this system will perish completely, or either this system will survive. And when it survives, the information, it's preserved. Therefore, if you have some sort of change in the information, or to call mutations or something, immediately, because there is a rule for determination of the structure and the function, immediately structure and the function will change as well that means you have a new situation or new structure and function will have to resist to the environment. And again, if it resists to the environment, it will be preserved again in the informational source because the function and structure preserve this information. So you have basically a loop of information, rule, and structure which preserve the entire system. So here is the important moment. This is a definition not just for life. It's the same definition is for Darwinian evolution. Meaning the Darwinian evolution and life are absolutely identical things. They are the same thing. So,
okay, can you give me a clarification when you say Darwinian evolution? I take that to mean natural or sexual selection.
It is natural selection. Definitely. As I explained here, you have environment which for somehow interacts with the function and structure and so on. But in this environment, the entire system may perish completely. Right.
And one other thing, do you mind, how are you defining information here?
I will go to this point later, but this is some sort of, let me say, strings which have a function, so execute some specific function. And the next slide will be a little bit more clear. So pay attention about the definition of life, which is absolutely identical to definition of Darwinian evolution. So life or Darwinian evolution should be a dynamically stable system where an information determines its own supporting structure. So that is very simple, but also it's giving you some idea for the actual mechanism which you're supposed to looking in the origin of life. And also, this is a moment where you are capable to upgrade the system. Without this information function system, you are not able to upgrade the system at all. Which means at the beginning of life, when the life started, this is the only mechanism where you can expect now to have a complication, adaptation, and upgrading of life. Without Darwinian evolution, without this mechanism, it's not possible to have anything more complicated. Or to say in another word, everything that it's assembled as a structure will be assembled again with the same probability every time if you don't have this Darwinian evolution. But once you have this Darwinian evolution kicked out, this system, every time, next time, you may have slightly more or higher probability to have the same system to exist, to assemble itself. So this is the only mechanism which secure further adaptation of the system. So also this system information and function, I mean this particular definition, do not explain in any way how complicated it is. It doesn't involve anything about how complicated it should be. This system could be extremely simple and still to have the same structure with information and function. And that's very important. Also, this system do not try to take into account what is the building blocks of the system, because in fact, in the laboratory conditions, you still can make the same system artificially and to call it life if it's subject of the same information and function interactions. So But this is important. It's not about how complicated it is. It's about the system of interactions. And also another thing which is obviously here, when you're talking about origin of life, usually so far, the scientist was thinking about something like a graduated process, which goes from the bottom to the top, from simple to the complicated, chemical evolution, after that all complicated and more complicated and more complicated. But between one, for example, the chemistry, and between biology, you cannot say, it's very difficult to point and say, okay, this is life, this is not life. But that was the old vision. This definition here say otherwise. It says that you have a very clear, distinct moment when you can call it life. Once this system is formed, this system is fully capable to upgrade itself and to continue to adapt. Therefore, at the moment when you have formation of Darwinian evolution, you have life.
Can I get another clarification? I mean,
yeah.
Most of us learned in middle school that there are 7 criteria to be alive. Are you referring to that at all, or are you describing something else?
So the dynamically stable system, actually, if you think about this particular definition, the dynamically stable system where information determines its own supporting structure could include a lots of things, and could include everything that it's known to be important for the life, like replication, like metabolics, and everything. So also it's important to point out that this is a dynamically stable, meaning it's adaptive all the times. It still reacts to the environment, but it is adaptive. And this is clear because you have a positive feedback. The feedback actually, it's something which secure the next step, the surviving process. So everything that it's known to be specific for life could be incorporated in this function and structure.
So we're talking about like 2 of the criteria that viruses don't meet. As one, they're not in a cell, but I guess the 3 of the criteria that they don't grow, and they don't have homeostasis. But we're not talking about any of that here, right?
So here, by this definition, you can immediately say that the viruses are not live, but they have a potential to be alive if they're in the host cell. So only when the process of information is executed to function, which is only the stage in the cell, you have a situation when you can call it life. But this is only that moment. Outside of the cells, when it's kind of stationary, steady state situation, it's frozen and doesn't execute any of this function. So we can call it unlife or non-life or whatever you want. It's a potential for life, but it's not life. Also, the same is for the seeds. Actually, if you look the seed of the plants, they're in a steady state situation and just waiting for the right environment to grow.
I like the phrase unlife as opposed to undead.
Yeah, it's unlife. So it's different than that, because that has not potential to be life. So unlife has a potential to be life. So in the right conditions, it's just in a steady state situation. And that's also important to say. Let's go for the next slide, because now it's getting slightly more interesting. So here you have, based on the previous slide and the definition of Darwinian evolution, you can say that during the origin of life, the information, the structure, and the corresponding rule are locked in their origin. Otherwise, those three components are mutually existent each other. You cannot separate because they cannot exist independently. To say it, for example, information cannot be any strings of something, without having execution of some functional properties. It's not information, it's just junk of nothing. It's a random. So it must be functional, otherwise it's not information at all. From another way to say, the rule, in order to do that, the information must have some sort of rule to execute the function. So in another way to say, you cannot have explanation of functioning structure without explaining information how come to be. Or if you want to explain the information how come to be, you must explain the function, structure, and the rule to execution.
I think somebody has a question in the chat, and I'm not certain that it's been answered. So the question was, I think it seems like we're going with RNA first, and someone here is asking, what's your position on metabolism first?
I will explain that in the next slides. So
okay.
I'm coming in those because at the moment I would like to explain a little bit about RNA world hypothesis, and not because I'm a fan of RNA world hypothesis, but just to point out what is the difficulties in RNA world hypothesis. So, and I understand right now here, for example, from this slide, that many creationists might flapping their hands at the moment cheerfully and say, I told you, see how difficult is all of that. So I will say, just stay until the end of this presentation. You will see how simple is actually could be that. So you don't need to have complicated things to have those interactions. You don't need complicated structures to have these interactions. Okay, so now we are going to RNA world hypothesis. I would like to explain about RNA world hypothesis. Okay. Yeah, my dinosaur constantly is bugging me, so sorry about that. Now, the reason I'm trying to put in the discussion RNA world hypothesis is because this hypothesis was dominating probably about approximately 30 years, the field of origin of life. And I would like to describe this hypothesis and also to show what is the problems of this hypothesis, and also to go for the next set of hypotheses, which is the RNA-peptide world hypothesis. So here is just the background about the information and function in the modern world, in today's of course, life as we know it. The information is carried on from RNA or DNA, but here for the simplification for the origin of life, we can say securely that the RNA it's coming before DNA, and there are lots of clues, there is lots of evidence for this. And of course, execution of protein and function structure, which are the proteins. So the idea is what comes actually first in that case. And RNA world hypothesis actually came exactly for the same purpose, to solve this problem and also to make it more simple. Back in time, the RNA world hypothesis was hypothesized by Gilbert in 1986. And he was encouraged heavily because of the discoveries of the ribozymes, which I just mentioned, the ribozymes. And the discovery of ribozymes received a Nobel Prize because this is a new type of enzymes new type of enzymes. So, and let's go on slightly more detail about RNA world hypothesis. Okay. So RNA world hypothesis postulated that first you have just RNA entity. And to exist this RNA entity, you have some self-replicating ribozyme. And this self-replicating ribozyme will continue to maintain this RNA entity. And these ribozymes will continue to maintain, and they will exist as RNA-only steps. However, during the process of formation of processes of mutations, not everything is perfect. Also to existence of amino acids around. Somehow you have a codon formation, which, by the way, according to RNA world, it's started to be independent from the process of translation. Somehow this process continue and form a translational system, which is the modern ribosome, but let me say it forms the proto-ribosome, the protoform. So based on this hypothesis, you have a huge process from RNA-only self-sustained system. You have a polymer transition that happened to RNA proteins self-sustained system. So this is the RNA world hypothesis just as a basics. However, in order to have a ribozyme, which is self-replication ribozyme, you must come here from very short RNA. And how that could be? According to Jacques Zostak and many others in Harvard University, they are very good scientists, by the way, but unfortunately, I don't think they are correct here. But anyway, so let's describe what is they thinking. So first, you have non-enzymatic replication, natural replication, which means if you have short RNA, so short RNA meaning the polymerization process could happen just naturally by adding the nucleotides on the top. This process has been shown actually to occur without any doubt, without problem. In the 90s has been shown that it's completely feasible as a process. However, it's mostly feasible and it's occurred for G and C nucleotides, but not for A and U nucleotides, for adenine and uracil nucleotide. So this process is basically blocked by adenine and uracil nucleotides. And according to the idea, in order to have process of developing of ribozymes and self-replication ribozymes, you need to have a high fidelity of this non-enzymatic process. If you observe the latest publications of Jacques Zostak, for example, they are mostly dedicated to the non-enzymatic replication. And he insisted that this non-enzymatic replication must be with very high fidelity in order to develop such a ribozymes. Otherwise, you cannot maintain these structures until the moment you have self-replication ribozyme.
I'm going to ask you to try to simplify that a bit, because we have some comments in the audience that they need a prerequisite course in order to understand this lecture.
Oh, okay, good. I'll do that. So sorry about that. My point was, it's very difficult to explain formation of such a relatively long ribozyme just from the point of short ribonucleotides, from short nucleic acids, from short RNA. So, and for the polymer transition, actually, to have a polymer transition, there is at least around 50 different hypotheses here. And most of them are very cool, actually. I like some of them are very kind of innovative to explain this polymer transition. But I don't think it's actually happened at all. So after all of that, because of many difficulties, as I explained, there's a lots of difficulties to explain the formation of self-replication entity of RNA, and because of not very good explanation for the polymer transitions that I tried to say, there is a really voice of doubt of RNA world hypothesis. And unfortunately, those kinds of difficulties in this hypothesis has been used a lot from the creationists to attack at all the origin of life. But even so, I think there is a solution, which is not RNA world, but RNA-peptide world hypothesis. So here I can say it's a very important to mention about the so-called rule of continuity. Meaning, if something is very important in the beginning of life, it still will be important. It's very difficult to read out of this. It's like something, let me say, like a genetic code. If you already set up a specific codons for specific amino acids, amino acids will go in a specific place, now it's very difficult to read out of this code, because now everything will become completely gibberish and will not be survival. The survival will be difficult. I want to say that here in this analogy is just the mill, process of mill of the grain. So you have at the very beginning in the prehistoric time stone 1 and stone 2, which continue to develop itself and evolve. We humans evolved that in a much more sophisticated system, going from different power sources and so on. But at the end, you always have the same principle, stone 1 and stone 2. It doesn't matter how complicated it is. You always have the same system, stone 1 and stone 2, to mill down. And it's all about this self-replication ribozyme. So far, never been found ribozyme capable of self-replication. There is a different type of ribozymes, but not self-replication ribozyme, which absolutely point that actually self-replication have never been occurred at the beginning and after at all. Why I'm saying this? I'm saying this because it constantly, a very high grade scientists continue to say self-replication, self-replication, self-replication. I don't think that ever happened at all. However, if I don't think that never happened, I should provide explanation what actually happened. So around 2015, one of the pioneers, like Williams, Professor Williams, he published a paper about that the ribosome challenge RNA world hypothesis. He is one of the pioneers which he postulated that actually you don't have just RNA, but you have something that interacts with RNA and the peptides at the beginning. So it's called RNA-peptide hypothesis, RNA-peptide first hypothesis. And it seems to me that we are talking that RNA first and peptide first, both are first. And I will explain just in the next slides how that happened. So it's a dance for two. It's not single dance. And that's for sure does not involve self-replication. Self-replication is not important for that process. So I'm going right now deep into the hypothesis, which is trying to describe all the process. By all of these introductions,
I'd like to throw one other thing in there. You said that self-replication was not necessary when you talk about the RNA and the peptide. We do need to, for those who don't understand it, there is self-replication for RNA at least. So we're not saying that self-replication doesn't happen. In fact, it does. It's just not for everything you're talking about.
Yes. Okay. So what I mean, self-replication has been achieved in the laboratory conditions. Self-replication ribozyme has been synthesized in the laboratory conditions.
And
So
when it's been synthesized, when we've demonstrated in a lab that there is self-replication, that means that there are chemical conditions under which self-replication occurs in nature also. And I just want to specify that because there are people to think, well, if you need a laboratory to do it, the reason that we need a laboratory to do it is because we're trying to look at it under a microscope, and it's hard to look at specific conditions, especially those of a prebiotic Earth, at any point under a microscope. So we have to simulate that.
So what I'm trying to say, okay, you can make in the laboratory almost everything from ribozyme in terms of activity and functions from ribozymes, and there has been done a lots of laboratory ribozymes for almost everything, including self-replication ribozyme. What is my point here? That naturally, in origin of life, there was not a process of self-replication at all on the RNA level. That's what I'm trying to say here. RNA has never been self-replicating naturally in the nature. So it has been not, there's no such a evidence from the nature, normal, natural process, which is self-replication of RNA. However, self-replication as a terminology exists because the cells are self-replicating. The entire system, biological system, are self-replicating.
I can cite a study that did show self-replicating without an enzyme.
No, this is a replication process, non-enzymatic. Yeah, there is a that's why I mentioned Jacques Zostak experiments. You can have a replication, non-enzymatic replication of RNA, and I completely agree with that. There is no doubt. It is shown by Jacques Zostak. You can use, for example, something like RNA-like, not exactly the same structure, but RNA-like RNA, and you can achieve all 4 nucleotides of replication, which is non-enzymatic replication. There's no enzymes involved. You have just primers and nucleotides in specific conditions, and you can achieve a replication. But this is not self-replication. It's not like you have enzyme that replicate itself, and so on and so on. I think this is one of the very important misunderstanding from the perspective of origin of life. So the very first misunderstanding was that life began and after that evolution kicks out. No, this is a misunderstanding. Life and Darwinian evolution are the same thing. At the moment when you have Darwinian evolution, you have life. There is no such a thing that life started and Darwinian evolution after that kicks out. This is the same identical process.
Well, I have to clarify on something because evolution, and when you talk about Darwinian evolution, you're limiting it only to natural selection. Natural selection can apply to other things, but life or evolution is descent with inherent genetic modification, and so we have to have the genome before we can have proper evolution, biological evolution.
You
Anyway.
have to have information source. Yes, I agree with that completely. And there's no any doubt about that. Of course, as I mentioned in the definition of life, that was one of the reasons I pointed so well this definition, is because you have to have information source in order to have Darwinian evolution. There's no doubt about that. For example, Paul Davis and Sarah Walker, they published a paper, I don't remember when it was, but a few years ago, and they said, okay, the life is information management, which is absolutely accurate. That is what it is. It's information management. By saying information management, however, you are not able to describe the actual mechanism in this way. So when I say that yes, information must be there, you have to say how this information interacts with the function and the structure, the actual structural component. And this positive feedback that I described is absolutely important for having Darwinian evolution. So Darwinian evolution and life are identical. They're the same thing. So here I'm trying to present the Earth conditions, primordial Earth conditions, just roughly without any details, of course, but you can expect lots of cycles, and that's a very important cycles in the primordial Earth. There is a wet and dry cycles, there is a hot and cold cycles, which are absolutely important for life to begin. And this is some people don't realize that you cannot have steady state situation and life to start. It's important to have different conditions and cycles like this. Earth during that time, first of all, Earth was slightly smaller at the beginning, and the moon was approximately 8 times closer to the Earth. So I would like everyone to just picture this situation. Imagine a moon which is 8 times bigger in the sky. So how cool is that to stay and look the moon? Just if you stay there, you can even see with naked eye. You can see how it's slowly moving around the Earth. What does it mean? That doesn't mean you have also a huge tides, enormous tides, meters high, many huge tides, which mean, let me say, tide from Atlantic Ocean to the Chicago, for example. Such a huge tides. And that's important. Why? Because in order to have a polymerization as a process, and this is actually what has been shown from David Demon, in order to have a polymerization process, you have to have a dry and wet cycle. Why? Because when you have monomers, to polymerize this monomer, you remove water. So that is the moment where the dry cycle is including. So you need to have an almost dry moment when the polymerization occurred. And it shows experimentally, actually, many times. So this is one of the contribution of the moon for origin of life. So let's go for the next slide.
Yeah, I just wanted to throw in, I'm still going with like a middle school level definition of life, which requires homeostasis, and we haven't gotten to there yet. And I'm also going with the what I think is the most basic definition of evolution, which is in descent with inherent genetic modification. We don't even have proper genetics yet. I mean, we technically do with RNA, but I don't know if you when you say that origin of life and evolution are exactly the same thing, you are talking about natural selection, and I agree, natural selection alone does apply here. But in biological evolution, natural selection is working on the fundamental process of genetic drift, and we can't even come to a point of genetic drift yet.
Here it's a more so the definition that I provided here, it's a kind of more not just general, but it's more detailed to the mechanism, and also it doesn't matter. Are you going to have, for example, a huge genome or small genome or any kind of genome? You have information there. And this information is good enough to trigger the positive feedback for whatever it is there. So the selection, I'm calling the selection mechanism Darwinian evolution here. So, I mean, that probably it's more. And once you see what the information is just after a few slides later, so you will see exactly what I mean, the very first information, because it must be very simple. If it's not simple, you cannot start. I mean, it is one of the default importance requirements for origin of life. You have to explain it as a very simple, probable way. If it's not simple, probable way, you have a very high, I mean, huge problem. It is huge problem if it's not simple enough. So here, for example, as I mentioned before, you could expect in the prebiotic conditions to have a very short RNA. Very short. I'm not talking for long RNA. I'm talking something from 2 base pairs to 5 base pairs or a little bit more. This is kind of good enough to have a process of hybridization between the RNA. So if you have short RNA and heat this RNA, what will happen? The 2 strings will melt down. And this is a denaturation of RNA. And that happened not just in RNA, it happened in every nucleic acid, DNA or RNA, or even hybrid. It doesn't matter. So, and at the moment, we see the same phenomenon in every PCR at the moment. This is the principle of the PCR. So you heat the DNA, and it's melt. And when you chill it down, actually hybridize, the primer hybridize and start the PCR reaction. So I even don't need to prove that it's happened. It's everyday life in the laboratory. So, and what is happening in the high temperature, you have mostly single-stranded RNA, and once it chilled down, it's hybridized based on the base pairs conditions. So I think it's very simple to understand that.
So I do think, like, and it is a relevant question to this idea, but during high heats, and especially like in an aqueous solvent, would it not be possible, like, for this string of nucleotides to decompose via hydrolysis, or?
It depends on the temperature
like
and the salt. And of course, the salt conditions matters to the moment when you have complete denaturation.
Right.
And the actual temperature matters. Usually when you set up PCR reaction, you have a melting temperature around 55 degrees for about 20 nucleotides primer. So that's the usual setup of PCR. So of course, it's not the same every time because you have different salt conditions, you have different nucleotide composition, which makes kind of variations, different variations of the process.
My point in bringing that up would be that the ocean, especially in the early Earth, is rather basic in terms of chemical conditions. But so would this process be occurring, say, on like some mineral surface, or like what kind of surface would be ideal for this process.
So,
too.
I mean, I forgot from previous slide to explain that you're supposed to expect some sort of compartmentalization processes. I even didn't, yeah. I even,
oh, go ahead.
sorry.
Could we explain that last diagram, those a little bit about those little figures, about the loops and the little 3 anticodon things, maybe for the people that don't know? Is that
After the definition of Darwinian evolution, you mean?
no, the last figure that you were on? Basically, I'm not entirely sure. Where was it? Go to the page that you were just on a minute ago.
Oh, go
with the
back.
little RNA strands. Yeah,
This one?
and and you've got like those little brush looking things are like anticodon relationships between 2 RNA strands, right? Correct?
Oh, okay. I
Yeah.
understand.
You know, and the loops, maybe explain the loops a little bit and how those happen in RNA.
Yeah. Okay,
You
so
know?
well, if you have a RNA, which of course, if it's longer, will make a secondary structure every time. Of course, as long it is, more loops will happen as a secondary structures. Shorter RNA doesn't have too much loops, maybe 1 or 2. That's it. And it shows hybridization process, which it's not referred to any codon or anticodon. It just statistically interacts based on a nature itself. When you have 2 or 3 common nucleotides, it's happened to be this with Watson-Crick base pairs. So if you have, for example, CCA, from the other side, if it's happened to be GGU, you will hybridize that in a lower temperature. And that's kind of absolutely natural. It happened very well every time. There's no doubt about that. So I'm not referring here right now as a codon or anticodon. I'm just referring hybridization process. Remember, I'm not trying to create life here. I'm trying just to observe the nature, how it behaves. I don't have idea how life began right now here. I'm just observing what happens. I will tell you later how life began, but at the moment, I'm just observing the natural process. I'm not telling the nature how to make life. So trying to be more like an objective here, not subjective for this. So any questions about that? Because we need to cover several slides more. And now gets more interesting, actually.
Go on.
So let's go for the next slide. Before continue, I have to explain just very briefly, which the reaction which I'm called the most important reaction in the universe. And this is the aminoacylation of RNA and amino acid, which it's happened on the ribose of the RNA with the amino acid. It's a aminoacyl group which interacts with the hydroxyl group of the ribose of the RNA. And this is a natural process which today it's happened over the tRNA, on the top of tRNA, where you have CAA end, the acceptor end of the tRNA. So aminoacylation, it's occurred by enzymes which is called aminoacyl synthetases. So those enzymes actually are doing those processes at the moment. However, I just want to point here that it could happen as well. It's happened without any enzymes. It's a natural process. This is aminoacylation process. You don't need to have enzyme to happen. The difference will be only that it's not specific. So any amino acid will interact with any RNA, There is no any specificity here because the specificity comes from the amino synthetases. Dr. Aron, any questions about that? So
I was just looking for my mute button. Don't call me Dr. Aron.
So the reason that I'm calling this most important reaction of the universe is because this is the reaction which gives a link between the information and the function. That is the actual chemical reaction which links both. Without this reaction, there is no way to have the life as we know it. So that's why I'm calling the most important reaction in the universe. That doesn't mean there's no more important reactions. There's many important reactions, but this specific reaction will give a links between the information and the function.
Well, I have to confess that, and it's more embarrassing that you called me Dr. Aron a minute ago, but I have to confess that my understanding of microbiology and chemistry, in fact, is so low that I wouldn't be able to tell you if you were wrong. So,
well, I put that from the books. Actually, this is a picture from the book, and everybody in the school knows about that, which is study molecular biology.
Maybe.
It is.
This brings back memories for me, particularly.
For those of us
so
that
from this slide, what is important just to know that amino acids it's attached to the actual RNA. That's important. So it's a called process of aminoacylation.
Yeah.
And here is just briefly to say what we can expect. What we can expect for the abundance of the amino acids in the prebiotic conditions, and that comes from a very detailed analysis, of the prebiotic Earth simulations and asteroids compositions to have idea what kind of amino acids you can expect to have in these conditions, prebiotic conditions. So you have approximately 7-8 amino acids, but what is also important, they're in a very different abundance. So the most abundant amino acids will be glycine, alanine, because those are most simple ones. And some of the amino acids will be 100 times less than glycine, for example. So that's a very important to keep in mind as well. So that's the reality you can kind of extrapolate from the experiments in that case. So here I have the same cartoon that I presented for the low temperature and high temperature, but this time I added the amino acids. So now you have the same process of in a high temperature you have melt down the RNA, so single strand RNA. In a low temperature they start to hybridize. But some of the RNA, short RNA, actually are linked with amino acids. So what happened in this completely natural process? Let's assume that you have two RNA that are hybridized completely naturally. On the end of this RNA, they might have amino acids. And just imagine also, it's quite natural to assume a dry cycle. In a dry cycle, those amino acids can close together just because the RNA are also close together. Because of this, you have a peptide formation, and you might have transfer of the amino acid on the one side or another side. In that case, completely randomly, you will have dipeptide, dipeptide, or tripeptide. And that process will continue up and down. Doing up and down in the temperature and dry and wet cycles, you can expect to form hybridization-dependent peptides. And this is a very short peptides. I'm talking about 2, 3, 5 amino acids, no longer. So that is the process of formation of hybridization-dependent peptide. It's a quite different from just smash the amino acids together and make a short peptide. It's not the same. These peptides are synthesized because of the close proximity that has been created due to the hybridization process of their RNA. I hope I was kind of clear what I mean.
Yeah, just to make sure these little circles and squares and triangles are amino acids.
Yes.
And the lines.
To say.
Yeah.
These small triangles, it's just a representation of different amino acids. Yes.
Yeah, and the other part was that there were some amino acids that are they're much easier to make, so they'll be more common. And those actually are the statistics of that is kind of what went into the genetic code. And that's kind of what this shows, that there's that we don't need 20 amino acids. And we don't, and maybe not even the amino acids we have today. There were some differences, but
but for the level of hybridization dependent peptide formations, so you have a short peptides, and this is what you can expect to have in those stage of peptides. Yeah. So these peptides contains, so those peptides are not coded, because if you think about every amino acids which is around, it's completely likely to interact with any RNA. That's why you don't have a process of coding yet. So the coding will come later.
Yeah, and the other thing is those very, very early amino acids, they represent, I believe, all 4 different kinds: polar, non-polar, charged, and negative and positive.
I understand your point, but that's not important right now.
Okay, so sorry.
Don't complicate.
I'm okay.
So if you complicate too much the picture, nobody will understand.
I will shut up now, Dimiter.
No, it is okay. So just I have a parrot problem, a dinosaur problem here.
Okay.
Just 1 second to remove the dinosaur that binding me.
Oh, the dinosaur's after you again. I have a carnivore that would love to meet your dinosaur. My kitties drag in dinosaurs every day, and they let them go.
Coincidentally, my dinosaur might give your carnivore a run.
Oh, really? You got a big one, eh? I would love to say. I would love to see him. I'd love to see the expression on my cat's face when he saw a big.
Oh yeah. Well, he'll be back anyway, but for now I'm just removing it. So that's what you can expect. And again, think naturally. I don't want to make life here for sure. I'm just thinking what will happen. So you have the peptide, just formation of peptide, which is non-coded. Second of all, the composition of this peptide will follow the abundance of the amino acids, and that's quite logical. If you have more glycine, of course will be more glycine in the peptide as well. So like alanine and glycine. So, but some, by chance, of course, which depends entirely on the abundance, by chance, some of those peptides will interact with RNA. Why? Because they contain a positively charged amino acids like arginine, lysine, or histidine, for example. Those 3 amino acids are positively charged. And those amino acids actually interact because of electrostatical reasons, of course. They interact with RNA. And of course, this is quite clear. If the peptide contains some of those three amino acids will interact with RNA. That's why the peptides that contains some of those amino acids that facilitate interaction with RNA, we call it RNA binding peptides. Those RNA binding peptides are, of course, hybridization dependent peptides, as I explained before. Some of those peptides contains a second amino acid, which is also important. It's opposite charge, like, for example, asparagine acid, which is negatively charged now, but it's just trapped in the process, this random process of hybridization-dependent peptide formation, and it's happened to contain two amino acids. One is a positively charged, another is negatively charged, but statistically, let me say 10 times less probable to happen this than the other type of peptide. So one in a, let me say, maybe 10,000 or 100,000 peptides, one will be, of course, bridge peptide. I call it bridge peptide, and I will explain you why it's bridge peptide. So the bridge peptide, because contains another opposite charge, attract positively charged amino acid and facilitate. Now you have a process which brings close together the amino acids which is positively charged and facilitate process of interaction between RNA and amino acids and aminoacylate this particular RNA. So this process is called assisted. So it is assisted by bridge peptide aminoacylation, which is very easy to explain from the bridge peptide point of view. So you just have from one side attached to the RNA, from another side attract the proper amino acid entirely by, I will explain a little bit later again. So what is also very important right now is the peptides that interacts with RNA actually makes the entire system more stable. It increased the stability of RNA peptide complex. Those RNA peptide complex actually is more stable, and that's a critical. This is absolutely crucial for the life to be formed. And for the people that not believe me, this is a paper just came out this year that investigated from Professor Williams. He's one of the pioneers of RNA peptide world, which actually he investigated exactly that, how stable will be the RNA peptide complex if it interacts. So this is experimental paper with experiments showing that interaction between short peptide containing arginine, containing lysine, containing histidine are more stable. So, and he investigated also some of three non-canonical amino acids like ornithine or several others, but basically arginine is the top #1 stabilizer here. So experimentally proven fact that if you have interaction between short peptide and RNA, it's more stable. And that is the most important slide of entire my presentation. So here I will explain how simple is the formation of coded amino acids from non-coded. So let's assume that entirely by chance you have a bridge peptide assisted aminoacylated RNA. And at the same time, there is a much more randomly aminoacylated RNA. Of course, no doubt. So up and down cycle that I explained with the temperature up and down will form hybridization-dependent peptides, which will be at very different variants. Some of them, of course, will be exactly resembled with some probability of chance bridge peptide. But this bridge peptide will go back and facilitate another RNA to be aminoacylated on the tRNA. Because it's a more stable form, as I explained. There is experiment that shows it's more stable form. Therefore, the aminoacylated bridge peptide assisted RNA will be with slightly higher chance every time when you go in this cycle. So more stable form will keep in circulation those kinds of RNA. Not to mention that actually this arginine, for example, have a more specific interaction with GC-rich RNA. Slightly, it's better interaction with GC-rich RNA. So, if you go here from bridge peptide, feedback, aminoacylated RNA, go back to the up and down cycles of creation of new hybridization-dependent peptide, you slightly increase every time. You slightly increase the representation of aminoacylated RNA, which have the positively charged amino acid, and slightly increase the formation of bridge peptides because of the stability. It's getting more stable. So it's very important to figure it out where in this particular picture the information comes from. The information is the link between the amino acid and the particular sequence of this RNA. At some moment, having bridge peptide or peptides which harbor more which will interact more with RNAs will be every time will increase the number and the probability to occur. This is the picture here, right now, here, in that particular slide. You see the Darwinian evolution in action. You have information because of the link between the sequence and the amino acid. And also you have a structure, which is the actual bridge peptide, or any other peptide that interacts with RNA. It's so simple, but it's also Darwinian evolution in action. How many amino acids are here important? Just maybe one or two important amino acids. How long should be the RNA? Not more than a few base pairs or bases. So because this slide is very important, I will show the same slide in another way. So this is another way of presentation the same slide. Here, the bridge peptide is shown in position 1. The bridge peptide possesses or contains arginine and also aspartic acid. With arginine, the bridge peptide interacts with RNA and mostly GC-rich RNA. However, you can see here the electrostatic attraction between aspartic acid and non-aminoacylated free arginine here. This free arginine is aminoacylated because of the close proximity, and this close proximity actually come because of the electrostatic interactions here.
If I may, just to, because I might have gotten confused about this, my dog's been acting up here.
Yeah.
We're talking about how we now have the short links of RNA have been joined into the longer bridge in a more stable form. Is that where we're at?
Yes. So once you have interaction between bridge peptide and RNA, those peptide RNA system or complex are more stable. It will circulate in the environment longer. Therefore, will be involved in a hybridization-dependent process more frequently than the others.
And that's natural selection at a really low chemical level, right?
Absolutely. Absolutely.
Which is what we need in order to show the development at this early stage. So
that's so simple, and it's a very early stage.
I think everybody's buried in
that is the key. So this is the key, because you don't have anything that it's not explainable here. Everything is known. I mean, hybridization process is absolutely known. The electrostatic interactions is absolutely known. The arginine that interacts with RNA, it's so well proven. Hundreds of papers. Maybe not hundreds, but okay, good enough number of papers. It's all based on the experiments and data. There's nothing here, absolutely no one interaction here that you can say, oh, no, that cannot happen. But you have more stable form. So based on this Darwinian evolution in a so low level, you have accumulation of bridge peptides. And selection of GC-rich RNA. Both process will happen here. Selection of GC-rich RNA and accumulation of bridge peptide. Of course, you will ask immediately, so what happened next? And I will try to go for the next to show what happened. But first, I would like everybody to understand that moment here, this cycle, because that's a very important. So just to say here back in this slide, I'm calling this very first Darwinian evolution. I can call it Terra Darwinia. This is the first life. The only characteristics of Terra Darwinia is having Darwinian evolution. I mean Darwinian selection. That's the only feature of this particular simple system.
I think a means of maybe simplifying this problem might be to ask that, so this process, at least according to my understanding of it, appears to be biased towards the inclusion of more, say, arginine-rich amino acids in its sequence.
Yeah. Yep.
Since the more positive charges you have, the easier it is to stabilize the RNA.
Yeah, the aminoacylation of arginine will happen more frequently.
A developmental biologist tried to explain to me what I think is the next step on this, and he did a good job because he's a professional teacher and all that. But I understand there's still some questions in how we get RNA to be in its job of creating DNA, which I think is a different process than it does now. And is that where we're going next?
Yeah, that's further up the rope.
So the DNA is coming much later.
Yeah.
So in order to have DNA, okay, you may have here DNA as well, but this DNA, it's not selected yet. And also, if you think about the clue, all of the clues that actually point that RNA is prior the DNA in the evolution, there are several very clear clues. First of all, there is no DNA-dependent polymerization process without RNA. Every DNA-dependent polymerization process starts with RNA primer. And I know that because I'm coming from the from the DNA replication field of study. Also, the formation of the DNA of desoxynucleotides actually goes first to formation of ribonucleotide. And there is only one enzymatic step that remove the hydroxyl group and make it desoxynucleotide. So the desoxynucleotides comes from ribonucleotides, evolutionary. Not to mention that many metabolic precursors or important moleculars like ATP, for example, nut, fat, all of these are RNA based on a ribonucleotides. So we have enough clue to say that RNA predates DNA. So I mean, probably a little bit later I will say something, but by now this is very important to show that the formation of Darwinian evolution at the very low level, in a chemical level, it's not necessary to be so complicated. It's very simple. In fact, you have just bridge peptide, which is hybridization-dependent peptide, which interacts with RNA, and that facilitates the loop. Okay, here is just summarizing what will go next, because nothing is steady state. You always have mutations, always have structure. You already formed the Darwinian evolution in action. Therefore, you can think about this short RNA to be the future tRNA. Also, the bridge peptides and any RNA binding peptides actually can develop, and I'll explain a little bit later, in a more complicated forms with motifs and longer peptides, and which will continue to develop as a next level of selection and formation of longer peptides and proteins. Here, for example, on the right, you can see the interaction between aminoacyl synthetase and tRNA, which now became very specific to find the actual amino acids and to put in the right tRNA because it recognized the right anticodon. And the interactions as it was in this process are considered as a proto-mRNA and will form in the future the ribosome. And there's many steps from here to here, but that's another discussion which is also important. I would like to say that at that moment you have a separation of the functions completely naturally happened, and that will give a huge advantage, selective advantage. I'll give an example. If you have a village, and in this village you have only 2 cars, do you need double direction roads? You don't need double direction roads because there are only 2 cars. These 2 cars to collide each other, it's almost unlikely. You don't need double directional roads. If you have at that moment 2,000 cars in the village, or 200,000 cars of the village, now you definitely need double direction roads, because that's the way how you will avoid collision. The same thing happened here. You need a separation of the functions to happen, which will give more efficient processes. And that's make the situation really, that will have a selective advantages when you start to separate the functions. Initially, any RNA can be either aminoacylated or serve as a template, for example. But later on, some of the RNA will be selected to be more specific to accept RNA. Another will be more specific to start and to form the ribosome. And third will be, of course, with the function of mRNA. So those kinds of selections will occur naturally and completely understandable. That gives a huge advantage of the situation. A few things I have to say about the formation of genetic code. here I can present, this is the genetic code which is ordered from GC-rich to AU-rich codon. And if you look carefully, you will see very clearly that the very first GC-rich amino acids which have a GC-rich codons are arginine, glycine, alanine, and proline. All of this naturally have a function, good function. You can figure it out what kind of function that can have in a small peptides. And after that, you have middle GC-rich, mostly GC-rich and AU-rich, and after that you have AU-rich codons. And I have also lots of clue, which I don't have time to explain right now, that the first RNA is mostly GC-rich. And this is about the actually non-enzymatic replication, because non-enzymatic replication naturally occurred more frequently with G and C. That's why in this system you have natural non-enzymatic replication, which continue to maintain the very first round of the replication. Because RNA replication or replication is so important, at some point, a very short peptide can hold 2 magnesium atoms, and that can serve and facilitate the process of first peptide that is able to facilitate RNA replication. So it's much easier in this way to describe and explain the formation of the replication than having a long process from RNA-like moleculars and self-replication, ribozyme, and so on. If someone wants to discuss that in details, of course, I'm open for discussions. But right now, I will continue. So, see this here from this slide particularly, you can see also very interesting phenomena. Ordering the codons from GC-rich to AU-rich, actually, it's also chronological. You have a chronological assignments of the codons in that case. And guess what? You see a zonal distribution, like this is green or yellowish or oranges, green and orange. That zonal distribution is corresponding to the different classes. There is 2 classes, 1 and 2, aminoacyl synthetases, which shows a mutual interaction between the process of codon formation and evolution of the aminoacyl synthetases. So there, it is obvious that this zonal distribution shows particularly that moment. What I'm trying to say, everything starts to fit. Observation fit with the model. So codon formation is very well explainable by this model. And now that's important as well. So someone have a dog problem. It's fine, it's okay. What is the difference between short peptides and proteins? And that's, although everybody know it, every school boy know, I mean, every student know it well, it's not kind of taken in account. Short peptides interacts only with the amino acids that actually possess this short peptide. But there is no folding process too much. Whatever it's in the actual peptides, what kind of amino acids form this peptide, That is all about the interactions with RNA or with anything else, not just with RNA. However, at the moment when the peptide became longer, now you have a process of folding, formation of 3D structures. Once it's formed 3D structure, you can form a catalytic center. And then you can perform much more different and variety of functions because of the catalytic centers. Another way to say, a short peptide have a very limited functions. Proteins have unlimited functions. You can make everything from proteins, and you can make all kinds of functions by proteins. That is why the ribozymes was selected in the first place. Because at the very beginning as the process, you don't have too much functions with just short peptides. A few short peptides cannot account for many functions. Therefore, at that particular moment, evolutionary advantages, some of the functions can be carried on by ribozymes. And that's explained why you have ribozymes selected. Again, the model fits the observation. Okay, next. This is overview. What will happen to the formation of some of the LUCA, the last universal common ancestors. So this is a major kind of summary of the major steps, what will happen until the LUCA is formed. As I mentioned before, at the moment when you have non-coded HDPs to the coded HDPs, this slide that I showed you with the formation of Darwinian evolution, you start the Darwinian evolution. And from that moment, ever since, it's undergoing selection process. The short peptides continue to evolve in a small motifs domains. And at some point, you have a much longer peptides which actually started to fold and to make catalytic centers. This is the moment when you must have specific beginning and specific end of the protein. Otherwise, you will have a frame shift mutation. Therefore, you have a protein development from that moment. So first, the evolution of proteins goes first from short peptide, motifs, domains, longer motifs in domains, and proteins. And within this process, you have also formation of the genetic code. Something which I explain in details in my second paper, 2020 papers. Until the moment you have a proteins formed, you can call this this period of time RNA-peptide world. Because at that moment, there is only peptides and RNA interactions plus ribozymes. And I explained just before a few seconds why the ribozymes actually was selected in the first place. And during that process, also you have a ribosome formation, and predominantly grew the translation machinery was started to develop predominantly. That is the moment when you have fully developed proteins and protein enzymes, which take over and actually populated all the functions. I'm assuming the very first metabolic reactions happens exactly in the very first period of time when you have ribozymes, because some of the precursors are ribonucleotides. And of course, it's accompanied with increasing of the genomic size.
Before you move on to the next slide, I just wanted to comment that at the beginning of this, I mentioned how I had listed a number of different studies showing many of the incremental steps, whereas, the anti-scientists want there to be a chemical that goes into a machine that cranks out a rabbit. And I was talking about how there's many different stages to this. And what you're showing in this illustration right now, this 5 stages that you've got illustrated here, are all within what I was calling one of those several stages. So it's further divided.
Yeah,
into,
this
into
is
many
just
tiny increments.
the major processes. So you just follow the major processes that must occur in order to continue further and further. And that's a naturally explainable processes. In fact, this is a new world for study, for research. And here, exactly here in this slide, it's entirely new world for study, for investigation, for research. Because we don't know a lot of details here. We don't know, but this is the major processes that must happen anyway. So we just need to investigate how in details happened and so on. So next slide is sort of the same slide.
We're running up against 2 hours. How long, or how many slides do we have? We have
So I'm
more?
finished. I'm finished.
Okay.
So
Okay.
I'm finished.
Just
Just
want to keep everybody
not
happy.
more than 5 minutes. Not
Okay.
more than 5 minutes. So here is the same as I explained before for the LUCA formation, starting from bridge peptide, but this slide is more dedicated to the formation of genetic code and how the genetic code actually was developed in a chronological way. So the paper from 2020 explains this process very well, and I'm dividing this particular process until the LUCA formation in 2 major stages, pre-LUCA 1 and pre-LUCA 2. Pre-LUCA 1, you have just a few amino acids which is actually assigned for the codon, and pre-LUCA 2 is the rest of the stuff. So there are lots of details here, but I don't want to go in details right now. But you can go to the paper and you can see all of that, how it's happened.
Are we going to open up to questions then, or are we?
So let's finish with this slide and we'll open for the questions.
Okay.
So how this hypothesis actually could be assumed as a reliable hypothesis based on my criteria that I started with. So here, this hypothesis solved the chicken and egg dilemma. It's obvious you have the same RNA and peptide first. There is no need. I like that. So,
Does he want to get in here?
yeah, I think your doc do not agree with me.
Do not, no, he's the creationist. You got a creationist dog.
Yeah. Okay, so chicken and egg dilemma solved by this particular hypothesis because it's RNA peptide by itself. It is so, and basically say that there is no such a question like a chicken and egg dilemma. So justification for formation of translation, I described that stability drives the translation process. If you have stable, the codon formations, for example, and translation is a process because it's more stable process. The stability is the reason to have a translation as a process. And that was explained very well. So I also explained why the ribozymes are formed. Because initially you have just the short peptides. And after that, you have a longer and longer peptides. Needs explanation what kind of simple structure can start Darwinian evolution. I just mentioned that it's a few amino acids and very short RNA. In the process of, you have a process of up and down cycles, wet and dry cycles, heat and cold cycles. needs exploitation where the energy comes. So that is from prebiotic synthesis. Most of the components like nucleotides, amino acids, they come in an already activated form. In pre-symbiotic pathways, they are already activated. So it comes with additional residues or additional components. They are not in a free, simple forms. That's already pre-activated to go for replication. And by the way, metabolism, according to this hypothesis, the metabolism is formed as a way of independence from the environment, from the supply of the environment. So the environment has some supply initial, so you have very simple interactions. But once the Darwinian evolution occurred and continued to develop, everything that it's capable to produce by itself, its metabolism, and makes an independence from the environment, which is a huge selection. So that's what was important. So I'm open for question discussions, but I have to go.
We wanted to mention we wanted to mention that we're going to try to model some of this in computer software, and
yes.
And I have some choices that I'm going to make, but there's some need for people that can do graphics and maybe some way to display, the results of these models. But basically, we're going to try to develop a model that sort of proves or shows some of these concepts and shows the possibility of this increasing complexity, which is not going to be an easy thing. But anyway, if anybody out there is interested, I know you are, Andy. So on the chemistry part of it, not the programming part, but.
Oh no, I could ask 1,000,001 different questions about the model.
We'll discuss that for sure. Absolutely.
Yeah, well, the model is going to be informed by the chemistry and by the theory, and we have to get down to the details. It's not going to be easy. So anyway,
I need to throw out another comment too. That what he was talking about so far, it basically just the formation of RNA. We still have so many other stages after that.
Absolutely.
So, and when we're talking about, like when it's actually life, by, as I said, the standard definition, where we're all the way up to homeostasis. Right now, if we're talking about a LUCA, last universal common ancestor, we could have here the last universal common ancestor of all life as well as all viruses. So we haven't even gotten into the biospecific stuff yet.
Yeah.
And then there's the other thing that you mentioned is the lamellae or lamella. Basically, I think lipids and certain glycoproteins and carbohydrates form can form little chunks that other things are attracted to, and there you have a very, very degenerate cell that's not really a membrane, but it's just a way of attracting things together, and there are so many billions of steps, that we have to go from here to eukaryotic cell that it boggles the mind, but you got to start somewhere. And I think Dimiter has started at a very low level and shown a very basic, more abstract form of natural selection that works at, these very, very small RNA peptide levels. So anyway,
I would like to tack on something to that. When you say very low level, it might give somebody in the audience perhaps an impression that it's not significant. In biology, the lower the level, the more significant it is.
And that's the amazing. But the things that attracted me to this hypothesis is the fact that we have this generator with feedback that complexifies and also does a bit of natural selection at a chemical level, and that model is kind of what we've been looking for. That's that is the golden pot of gold at the end of the rainbow here. So a very appealing hypothesis. So
of course, as a scientist, I want to say that this is, yes, it is a hypothesis, but also it's based on everything that we know and experimentally we know and what we have. it is a way that definitely needs experiments. I know exactly what kind of experiment must be done here. Unfortunately, I don't have my own lab here to start and doing those experiments. But anyway, these experiments must be done in order to continue for the actual science. So what the experiments do? The experiments might completely disprove my hypothesis. And I'll be very happy with this as well. If someone is capable to say, okay, to put the finger and say, okay, that will never happen. That's why your hypothesis is wrong. I'm fine. I will accept it if the experiment shows, if the actual science, and you have a scientific argument for that, but not to say that, oh, that's not possible just because I don't like it. So I'm not too much kind of attracted. So the people should not fall in love with their own hypothesis. That's very important in science, because I see that phenomena over and over constantly in the scientific field. Some people so much are attracted to their own hypothesis, they don't want to look anything else. So
a very important point.
Yeah.
My personal preference is to work with two hypotheses at the same time.
Yeah, exactly. So any questions specific? It's getting exactly 2 hours. Thank you.
Yeah.
Yeah, I'm looking in the chat. I don't see any questions for you. I don't know if anybody else has picked up some.
I think everybody. I'm not sure why, but
it's just impressive that Anthony Maurice hasn't showed up and tried.
I know,
to,
and told us abiogenesis is false. That's all he ever says. He says it 1,000 times.
Unfortunately, I was looking for the keyboard warriors.
We got
actually,
we have.
we call I'll be back in 2
Joel.
minutes. In 2 minutes, I'll be back.
Okay, we have Joel in the chat, SFB mod, and he's we call it speaking in tongues, when they learn words and they string them together and they don't make any damn sense. And he's wonderful at it. He can speak in tongues with the best of them. The other one is John Mad Ox, we call him. And these guys, they learn enough about it. I mean, they learn enough from the Wikipedia to speak in tongues and make it sound like they're actually coming up with something useful. And it drives me crazy, because when you listen to them, or if you go check their source material, they're just full of shit. They haven't understood what they need to understand in order to make those statements, and it's maddening. But the thing is, I believe that they believe what they are saying.
Well, when they believe it, I would agree they believe it too, but they use a different definition. If I say that I believe something, it means that this is what I think is most likely true or closest to the truth, but I can't prove it. That's what believe means to me. Believe to them means an act of will, of determination, that you will convince yourself of something that is not evidently true. And yeah, they absolutely convince themselves of things that are not true. This is why they don't work with any hypotheses. They don't offer any way of testing it. They don't have any evidence to support their claim, and they will not ever accept any future observation, and they won't make a future observation that would disprove or falsify their own position.
Yeah, and that's what scares the hell out of me. And no, Joel, that's not hate speech. I just stated my opinion about you guys. And I mean, I have worked to learn this subject, and when I listen to you butcher it, what else am I gonna tell you, or what else am I gonna do but call you ignorant?
There are a handful of people in history that have come up with predictions. And, you know, predictions are like the most important thing in theoretical science. It doesn't make predictions, and can those predictions be fulfilled or falsified? Because we need both. And there have been predictions made that have been falsified, that were made on behalf of creationism. For example, Richard Owen said that when the first Archaeopteryx fossil was actually a fulfillment of one of Darwin's predictions, and Owen tried to counter that by saying that, you know, that one was found without a head. So if they ever found another Archaeopteryx, it will just have a normal toothless beak like any other bird, because it's just a bird. That's what he said. And then they found later ones, of course, that didn't have a beak, that actually had teeth, and refuted his prediction. But of course, he won't accept that falsified his prediction because he's a creationist and he does not admit reality. That's his game, to refuse to admit reality. Other people came along and said, well, you're never going to find, there's no purpose for a half a wing, so science will never find a fossil with a half a wing. And now we have buckets of Paravian dinosaurs that have half wings. So there's another one that's been falsified.
Yeah.
But they won't ever admit that it's falsified because they don't care what science explains. They only want science to not explain things. Because if science knows things, that means that they don't know what they want to know. Or they can't say that they know what they don't really know.
Well, they think scientific papers are a source of sound bites.
Yeah. So anybody in the chat here, I present a question for our speaker, please. But otherwise, if you're going to just say that, well, that was disproven, explain how it's disproven, because I know you can't. Give me the citations that show it was disproven, and also show what hypothesis you're working from, what predictions it makes, and how do we falsify that? Because if you don't have any of it at all, then you're not playing the game of science at all. You're just sticking your fingers in your ears and beating your head against the desk.
Yeah. So I'm gonna. We have a fellow. We have a molecular alchemy. He's also a molecular biologist, PhD. I think double PhD, who works in cancer. He's not here today, but Leo wants to come in and say hi. So I'm going to send a short invite because I think we're wrapping up. So,
yeah.
But anyway, we have a group of people that we put together that kind of know what they're talking about, and we're trying to do better content on this channel. Though my production values are crap. The name of our production company is Production Values Are Not Us, basically. Let me see it. Okay, I sent something to Leo.
Yeah, and Andy, is it?
Yes, I usually go by Immutable, but I go by Andy.
Yeah, I'm glad that we had somebody in here that at least understood college level chemistry.
It took me like a few times, like smashing my head against the desk to get a hold of Dimiter's paper and to fully understand it. Because I will say, right, if you try to read that paper and don't have at least orgo or some level of like biological chemistry in your background, you're going to have a nightmare dealing with that paper. Needless to say, chemistry, as somebody can tell you, it's not a simple field. It makes sense, but it's you sacrifice brain cells in the process of that of learning it.
Here goes the neighborhood Leo's in.
All right. Well,
anyway,
I want to thank our speaker for being on and for the work he's done. I understand he has a number of published papers, and now he's. I guess Dimiter, you're working on. You said you're going to perform the experimentation to do another published paper on this hypothesis.
So I'm working on the third paper.
I can't hear you, sir.
So I'm working on the third paper, which is already it's more about the description of Darwinian evolution and what we can call life. So the first part of my talk, it will be published very soon. Yeah. I'm starting next week pretty much another, just molecular biology in the laboratory, but totally different from origin of life. I'm starting to work with the stem cells research because that's why the people pay for at the moment. So for the purpose of experimental work, the experimental research for origin of life, I hope I can convince my friends from back in Bulgaria. They can do something preliminary research, preliminary experiments, and they can do it because I cannot do it at the moment with here. So, and that's what the actually is. There are lots of people that have some money for. Not lots, but a few laboratories that have money for origin of life research. But they, for example, Harvard University. They're doing experiments dedicated to this non-enzymatic replication, but I think this is dead end in this particular research. But anyway, so that's what it is.
I did have
Okay.
a question for Dimiter.
Yeah.
And I've asked some people that have credentials in the field this question as it concerns abiogenesis, that they say there isn't just, if we look at how, when life first developed on planet Earth, it wasn't just a replicator first scenario, or just a metabolism first scenario, or just a membrane first scenario, that because even on the Earth throughout its surface, there were differences in temperature, differences in chemistry, differences in energy, and that it was likely a mixture of all of these different models. So in certain areas, replicators might have been developing first, but in other areas, metabolisms might have been developing first. Is that correct, or is it likely that it was one of those models all over?
I could answer that one.
Yeah, both of you can answer. That is just fine.
So what I just want to say before Aron, you don't need to diffuse too much the question. You cannot diffuse it too much. I mean, you have to explain somehow how Darwinian evolution occurred. If you don't explain the momentum of formation of Darwinian evolution, you don't have life. Nothing else can happen after if you don't have Darwinian evolution. So metabolism first hypothesis, they are very good of explanation of, for example, prebiotic synthesis, some of the conditions which prebiotic Earth could have, and this is very good to explain how was look like some of the environments in the prebiotic Earth, but cannot explain the formation of Darwinian evolution. So all of these processes and genome era cannot be explained by metabolism first hypothesis. They can go to some point. Also, RNA world hypothesis, which continuously to say self-replication, I don't think it's ever happened at all. Nothing like self-replication, and I'm talking self-replication only for RNA, for polymer, not for the cell by itself. Of course, cell by itself is self-replicating. But when I'm saying about RNA level, there is nothing like self-replication ever happened. This is, I think it's a wrong concept, and should, I mean, one of the reason for we don't have such a breakthrough in origin of life, unfortunately, one of the reasons is the domination of RNA world for 30 years.
I'm gonna have to share a paper with you that does say that RNA can be self-replicating, that there's certain chemical environments where RNA will self-replicate even without an enzyme.
That will be nice. I will be very curious.
I had a question on that, Aron, and I don't know if you'll remember the names of who wrote it, but is that paper, do you know by chance if that was the one that was done in 2009 by Gerald Joyce and Tracy Lincoln, where they showed that what is it? So I think it's like short strands of RNA can self-amplify utilizing oligonucleotide substrates.
That sounds right, but I'd have to go. I have to go search through it because I have a document that I could find that in.
Because
unfortunately, I don't have access to it at the moment.
Right on. Because if it is, that is a very interesting paper, because they also showed that I think that a genetic recombination can occur through certain Watson-Crick pairs during this process, and that can lead to a sort of selective process whereby these RNA substrates will compete for those oligonucleotide substrates in their self-amplification. So it sort of can show this natural selective process that Darwinian evolution is based on.
Yeah, and when we say Darwinian evolution, because I saw a question in the chat that was confused about that. Remember that Darwinian evolution, whatever it means to the creationists, to the scientists, it means pre-genetic, because the Mendelian contribution became the modern synthesis rather than Darwinian, and so it was no longer Darwinian. So when he's talking about natural selection, you can have natural selection and it not be what we now understand today as the modern synthesis, or the modern evolutionary synthesis, which is based on genetic, excuse me, damn phone, which is based on genetic drift, as I said, the constant mutations. But Darwin didn't understand anything about mutations. He didn't know what the variation was caused by, so he would accept any cause for variation. and thus we can apply natural selection even when we don't have functioning DNA.
Yeah. Okay, guys, what else?
I don't know. Is SFB mod? Is Joel? Is he still raging in the chat, or did he leave?
He's raging in the chat. I'm still trying to figure out how to have a talk with that guy.
Yeah, well, he won't have a talk with. He absolutely fervently refuses to have a talk with me ever since he tried to tell me that supernatural numbers prove that supernatural things exist, which is as idiotic as saying is that imaginary numbers prove that imaginary things exist.
So,
and John Maddox, I like to make fun little names for them. One of them is John Quantum Tunneling and Respiration Maddox, and another one is John Wave Function Collapse Requires Consciousness Maddox. Won't talk with me since I came into one of his streams. It was actually an after show of the debate you had, Aaron, with T-Jump against Cliff and Stewart on the Soul. You guys did beautiful, by the way, where I educated the entire stream on quantum mechanics because none of them had any fucking clue what they were talking about, and now none of them will talk to me at all about science.
So tomorrow we're going to be. I don't know if you're aware of this John Maddox, Matt Dillahunty thing, where he was mispronouncing Matt's name and insulting him. God. He was going to debate him, and then Matt said, "Fuck you, I'm not doing it."
# John Maddox is a grifter.
Yeah, it was funny. The whole thing. I mean, sometimes these creationists do the funniest things. And we love them.
Oh, you know what we need to do, Speed? We need to start a new thing. Creationists say the darndest things.
Well, I like creationists speaking in tongues because that's when they're talking technical. So anyway, you guys want to wrap this up and maybe go backstage for a minute and say goodbye? Or
okay.
Yeah, I'm going to have to bail. Thank you for having me on. I'm glad it was somebody that understood chemistry better than me, because there were sometimes when we were in the weeds, when I realized that, you know what, this guy could be misrepresenting all kinds of shit and I wouldn't even know.
That's the beauty of science. But thank you so much for showing up, and would love to have you repost this on our channel and give us a little shout out.
Okay, and Nesslig says that he has a question, and I guess we didn't get to it. So quick scan. Where is his question?
Is this it?
I don't see it.
Question. For a moment, he dissed, quote unquote, dissed I think this is for Dimiter.
Yeah.
Dimiter. Question. For a moment, he dissed metabolism first, but could metabolism first not be a previous stage that supplied the materials for the RNA peptide world he is proposing? I don't know if he dissed it so much as explained that a metabolism first scenario only gets you so far, but it doesn't get you all the way, whereas a replicator first model gets you significantly further along that process than a metabolism first gets you. Correct me if I'm wrong, Dimiter.
So metabolism first, it could be explanation of prebiotic synthesis, the prebiotic conditions, where you can, at some point, you have to come with something like a sequence that that could be rearranged in a information, but this sequence also should execute some sort of function. Therefore, metabolism first, it's a good start, but does not explain how the Darwinian evolution started. Until you don't have a system between information and structure and this feedback, you don't have this selection mechanism. If you don't have this selection mechanism, nothing can go complicated, cannot be more and more complicated, cannot be adapted, and so on.
Okay. I don't know. We got anything else? I think that's all we got.
Before you end the stream, tomorrow at Saturday night at the Atheist, and NathanArt was a witness to all of this, both on air and off air on Rage's stream last night. But yeah, Otangelo got his ass handed to him on a pretty little silver platter by both me and then Doubting Thomas off air. Oh my God.
Share me a link and timestamps if you would, please.
Yeah, we need to make that a regular.
I'll have the timestamps for me correcting him on a very, very small aspect of the Kalam cosmological argument, because I absolutely love destroying that, because when I was a theist, that used to be my favorite argument. And I didn't have enough time to parse out why both premises in that argument are actually wrong. Yes, that does include the first one, but he tried to go on about time being tensed and tenseless and how, well, we don't really know. And I'm just like, no, we've done experiments. We know for a fact that time is tenseless. And then off air, oh, fuck, that was bad.
We'll stay in touch.
When do I get to hear what happened off air?
I don't know if you know who Doubting Thomas is. I think he kind of stays more in the comments, but
Saturday Night at the Atheist?
Yeah. Me and Nathan, since we were there, we will be talking about that. So
where?
On this channel, it's called Saturday Night at the Atheist. It's every Saturday night. It usually starts at 9 p.m. Central Standard Time. And I don't know if you're Mountain Time, Aaron, or Pacific Time. Oh, you are.
Revolves around me, so I'm Central.
Saturday
why
night.
do I think in Texas is more west than what it is? I don't know why.
Saturday Night at the Atheist is where we kind of let our frustration roll, and it's irreverent as hell. And the more irreverent, the better. So anyway,
but I'll
send you a link, Aaron, if you want to. If I send you a link and you stop in, maybe we can address that part.
Yeah,
just for you.
Well, we'll be talking about it regardless, because I felt bad for him, but it was definitely needed.
I would also love to hear your attack on the Kalam, because it sounds like it's the same attack that I use, especially since you said both premises.
Yeah.
So probably the same, but I'd still like to hear it.
Yeah. I can send you the link to the after show that Rage had, and give you a few of the timestamps for that, or the timestamp for that. Yep.
Okay.
All right.
Okay, I'm going to end this and go back for just a second. Thanks, everybody, and we'll see you later.
And as always, Santa
Thanks
Amelia
again,
Park.
Dimiter.
Yeah.
Thank you very much.